sort of maps out where we should be applying our efforts and our research dollars in terms of trying to tackle and conquer this disease. So I want to start off, just change. Why is it important for us to understand the genetics of ALS and really genetics of any disease? Well, I think that the first thing is it tells us about how the motor neurons are dying. It gives us an insight into the biology and the pathways which might be important in sustaining just those motor neurons. And if we know what the important pathways are, it means that we could actually maybe do something to try and support them, keep them going. The other aspect of this, and I think Lucy showed you a lot of very nice pictures of different animal models, and I think that really highlights the plethora of animal models that can be made nowadays. But I think that the key here is that once we know the gene, we can make animal models. And why is that important? Well, it's important again because it tells us a little bit about the biology of the disease and what's important to keep in terms of keeping the motor neurons alive. But even beyond that, it provides us with a vehicle for testing drugs or potential therapeutic agents. Because of course you really don't want to be testing drugs for the first time in humans. You want to be testing, you want to get some hint about the toxicity of the particular agent, and you want to get some hint about whether the drug is actually going to work. Because otherwise you don't really want to bring it into humans where it could potentially, be detrimental as much as anything else. So I think that animal models are crucial with regard to that. And then of course it gives us therapeutic targets. The cell, the motor neuron in particular, it's a big place. There are many, many, many things going on, cellular processes going on inside. And if you're just trying to blanket bomb it and go in and change everything, that's hard to do. But if you know precisely what's going on within that cell, then maybe you can actually target and you have a better chance of changing it around and slowing the progression of disease. Now, what I want to talk about is about chromosome 9, the chromosome 9 locus. Now, this is a story. Basically, I'm going to tell you a story about how the area, the region was first identified, how our lab, my lab, got involved in it, and how we finally managed to crack it and find the underlying mutation. It's a bit of a story, and take you along. It spans over about 5 years at this stage, so it'll take a few slides to go through it. But basically, this locus on chromosome 9, remember we all have about 22 chromosomes, including sex chromosomes, and we're talking about the 9th chromosome. And on the short arm of 9th chromosome, there was identified back in 2006 a region of about 7 million base pairs. And that was identified based on 2 families. One from the Netherlands and one actually from Scandinavia, which you will see as I go through why it's so obvious that it was from Scandinavia. So it's about this, we knew that this region here on the short arm chromosome 9 was important. It was about 7 million base pairs in size, because remember our chromosomes are made up of these sort of base pairs and letters of the alphabet. There's about 7 million of them. Now just to put that into perspective, overall if you look at the diploid, Usually if I gave this scientist, they go, no, you're wrong. But I actually do think, and I think it's through a combination of what we call exome sequencing, genome sequencing, which is really becoming to the fore, and large-scale retrievals. And I do think that that is a realistic target for us. We know the pathway. It's not, I shouldn't really say this, but it's not rocket science, really. We know what we need to do. We just need to actually do it. We need to collect the samples. And there are a number of things. I know that sounds easy. This is what we need to do. It sounds easy. There are a number of interdigitating things that need to come together in order for that to work. First and foremost, we need you. We cannot do these genetic studies without samples. I can have the best machines in the world, the most expensive machines in the world. Unless I've got some stuff coming in the front door, it's not going to work. I think that the National ALS Registry, together with the pilot biorepository that they've set up, and also the Korean repository, which is funded by ALS, I think they're going to be key to this. And I think that the ALS Registry is really to be praised. They are really trying hard to gather everybody's information and to make it available to researchers. So I'd encourage all of you, if you have half an hour or an hour's time to actually spend that going on the website for the ALS registry and actually enroll it, because I think that that's a very powerful mechanism. The other thing that we need, and I'm going to bring this, I'm going to come back to that. I'm actually going to, the other interdigitating aspect of this is money. So we are very fortunate at the NIH to have deep pockets. We are funded by your tax dollars, and we are very fortunate. We've got great resources. But these experiments are very expensive. So even with all of that money, if it wasn't for the ALS Association, if it wasn't for Packard and other charitable organizations that are willing to fund our research, we simply would not be able to do that. And really, that is the truth. And honestly, you know, you guys, I know you will go out and fundraise for the ALS Association. This is what your dollars go towards, okay? It's not some pie in the sky. This makes a difference. Understanding this disease really makes a difference. The other thing I'm going to end with this point is the acknowledgement slides. Many people just put them up and list off all of the postdocs and so forth who did all the hard work. I can stand up here and look good, but they did all the work, and that is true. That's not the reason why I'm putting that slide up there. The reason I'm putting that slide up there is look at how many names are on that from all around the world. Gone are the days of the science being the permit of the sole lone scientist working late in his lab at night time. We are now into the realm of international collaborations. Without that, we cannot get together and get sufficient sample size. So I'm going to stop there because I know we're running out of time, and I'm happy to take any other questions you might have. Thank you.
Thank you, Brian, for a tremendous presentation. I know that you all needed to grab a coffee before the clinical trial session, but let's have at least 5 minutes or so of questions.
What is the comparison between the frequency of your repeat with people who have ALS or have developed it and those who have not?
That is an excellent question. We have screened, so right now, familial ALS, it looks like, depending on which population you're talking about, it accounts for between 40 to 50% of all familial ALS cases. If you look at the sporadic, it accounts for about 7%.
Yes, but
if you look, no, I'll get there. If you look at controls, and we've sequenced about 5,000 controls, we have found 5 individuals that carry the expansion but do not apparently have disease. Of those, 2 of them were actually Finnish individuals who died in their late 80s. One was in their late 80s, the other in their early 90s. So they're in their 9th and 10th decade of life. They were able to survive that long without developing disease. So we know that you can survive for that long without getting disease. In contrast, we also know that there are patients who have this expansion who get disease in their 8th and 9th decade of life. So those 2 came from Finland. There were 2 Germans that carried it, and there was an American that carried it, apparently neurologically normal individual. They were aged 45, and I think 62 and 61. So it's possible, given that the median age at onset is about 67, it's possible that they might actually go on to develop disease later on. We don't know. Does that answer your question?
The second question, will your talk and also will the paper from Neuron be on the ALS website?
I don't know about my talk today. There is a webinar, with Rosa, there is a webinar summary of this on the ALS. Lucy was kind enough to ask both Rosa and myself. Remember, Rosa Rabemakers was the other person who found this. So it's sort of a shared webinar between Rosa and myself, which goes into much more of the scientific details with respect to this.
And paper?
Well, remember the papers become, so NIH